Neurodegenerative diseases can stem from a multitude of issues, one of which is the presence of a misfolded prion protein within the mammalian body. Although prions are present within all mammals, the misfolding of the protein can lead to drastic and deathly results. A prion is defined as a protein-only infectious agent with a complex quaternary structure, which causes fatal brain diseases following silent incubation periods that can span a lifetime. Prions do not have their own kind of genome, but are instead an infectious agent with a gene that codes for the toxic part of the prion protein. The presence of the PRP-SC protein causes a correctly folded protein to become misfolded and harmful.
The diversity of prions could possibly account for the ability for some amyloids, which are proteins that fold in a way to allow many copies of the protein to form groups, to develop into infectious pathogens while others do not. Prions typically target highly defined regions of the brain. They lack nucleic acids and therefore cannot be determined using differences in the amino acid sequences of PRP. John Collins brings about the idea that prions may consist of multimolecular assemblies created by misfolded PRP monomers that form amyloids. This is an alternative hypothesis that has not been proven so far. Generally, prion strains are propagated into another distinct strain and can be expected to revert to a distinct strain if cloned. The type of strain is essential to what the prion can do, survive, and spread to in terms of host.
Diseases such as CJD and mad cow disease are caused by prion misfolding and propagation, as well as exposure to a certain prion. Misfolded prions can lead to toxic results, ending in death. CJD is a degenerative brain disorder that leads to dementia and is ultimately fatal. Mad cow disease is a variant of CJD that takes place only in cows but has the same end result. These diseases can arise spontaneously through infection or inheritance, but can go unknown for a period of time by existing in a silent state. Some prions can act as a template that are then biologically selected to be duplicated, kind of like biological pathogens by using PRP monomers. This suggests some sort of protein folding inheritance. Over time, this growth may manifest as some disease. The goal of this review was to see how prions that act as templates for other prions relate to other neurodegenerative conditions. Neurodegenerative conditions are thought to not be acquired from the environment, but if they are related to prions, it may change how we approach these diseases.
Prion-like mechanisms may potentially be used as treatment against other neurodegenerative diseases if in a non-toxic configuration, kind of like a reversal of the prion propagation. We don't really know enough about these disease-causing prions to be able to have an early detection mechanism. That way we could test potential reversal mechanisms.
Future work will involve connections between protein structure and protein-based inheritance, while working toward the ability to classify human prion strains as related to their molecular makeup and criteria.